In accordance with the customs of this university, I open this ceremony with the following words: Spiritus Sancti Gratia, illuminat sensus e corda nostra. Please take your seat. It's indeed my pleasure to welcome you all to the promotion ceremony of Mr. K Boahen. My name is René Bindels. I'm Emeritus Professor of Physiology at this university, and I'm today replacing our rector, Professor Jose Sanders. It's actually my job to take care that this ceremony runs smoothly and that all the opponents do get an equal time slot to ask their questions. And it's actually great that you all came to this ceremony in such a large numbers. And I also would like to obviously welcome the people who follow this ceremony via the live stream. Being here, you show your interest in the topic of today, genetic factors contributing to the immune response, but above all, you support with your presence our PhD candidate today. So I now give the floor to our candidate for the degree of doctor.
With the permission of the doctorate board, and in order to obtain the degree of doctor from Radboud University Nijmegen, I would like to defend in public my dissertation entitled Genetic and Genomic Regulators of Immune Parameters in Health and Diseases. The immune system consists of a complex network of cells and tissues which perform a crucial role in health and diseases. In health, it ensures stability of the body's internal environment. In the course of these activities, the immune system can sometimes malfunction, leading to various forms of autoimmune diseases. To understand the function of the immune system thereof, various aspects such as cytokine profiles, expression levels, as well as other markers are measured, and these are collectively called immune parameters. A fundamental feature of these parameters is their variability among individuals spanning from lower to higher levels. This variability is associated with individuals' propensity to diseases and also influences vaccination's outcome. Excessive release of cytokines, often referred to as cytokine storms, can sometimes lead to fatal consequences, as seen in viral infections like COVID-19. Trait variation can stem from genetic, non-genetic factors, as well as the interplay between these two elements. In the context of genetics, the first step is to assess the proportion of trait variation that is due to genetic factors, which is termed as heritability. Traditionally, family-based approaches such as the twin study design is being used. The ACE model, which makes use of monozygotic and dizygotic twins, is able to evaluate the share of trait variation due to additive genetics, common environment, unique environment, depicted in the PATH model as ACE respectively. Recently, methods that make use of live samples of unrelated individuals, such as linkage disequilibrium score regression, genome-based restricted likelihood methods, are also being used. Once heritability of a trait has been established, the next is to identify the genetic factors. Examining the DNA sequence of multiple individuals reveals regions where the nucleotide bases differ. This variation in more than 1% of the population is termed as single nucleotide polymorphisms, abbreviated SNPs. As illustrated, the first SNP contains alleles A and G. The combination of these alleles give the SNP genotypes. If A is, for example, less prevalent among the individuals, it is termed as the minor allele. The GWAS approach is being used to identify SNPs that are associated with complex traits and diseases. This methodology makes use of a substantial number of cases and controls. Here, the minor allele frequencies is compared between both groups. As illustrated in the last column, regions where the minor allele frequencies differ significantly between the cases and the controls are deemed as susceptibility loci. Quantitative trait loci mapping refers to the methodology when instead of dichotomous variables, continuous measurements are correlated with the SNP genotypes. A SNP is deemed significantly associated with a trait when there is either an upward or a downward trend among the genotypes. Conversely, when there is no distinguishable trend among the genotypes, the SNP is deemed insignificant. Cytokine QTL can be classified as either cis, that is when the SNP is in close proximity to the tested gene, or trans, when the SNP is situated distance away on a different chromosome of the gene. To date, the genetic basis of immune parameters have been elucidated, but mainly in individuals of European population. In Chapter 3, induced cytokine profiles were measured in an infant from Guinea-Bissau who were either received BCG or not. Substantial number of cytokine QTLs were observed between both groups. It was appealing to uncover genetic variants whose effects manifested only in the BCG group. Delving into specific, a SNP on chromosome 20 was associated with interferon gamma following peripheral protein derivative stimulation. However, this effect was entirely concealed in the control group. Likewise, in Chapter 4, cytokine QTL mapping were performed in adult population from Tanzania as well as the Netherlands. As indicated, a substantial number of the tested SNPs were insignificant in both groups, as depicted in gray. Notable cytokine QTLs, however, were identified in the Tanzanians, as depicted in green, as well as in the Dutch population, as depicted in purple. Excitingly, it is important to highlight the restricted percentage of overlap, less than 2%, of the genetic variant that influence cytokine QTLs between both populations. Beyond genome-wide comparison and delving into specific genomic regions like the toll-like receptor locus, two SNPs significantly affected induced cytokine responses, mainly in the Dutch population. Also, by looking at the linkage disequilibrium plot, which gives the pairwise association of SNPs in this region, a weaker as well as smaller LD blocks were observed in the Tanzanians compared to the Dutch individuals. From anatomy to physiology, we see clear differences between males and females. Also, in disease conditions such as autoimmune and inflammatory diseases, the propensity is shifted towards females, yet sex stratified analysis has received little attention. In Chapter 5, two population-based cohorts were used, and following protein QTL mapping, a stratified meta-analysis revealed that 67% of the SNPs that affect inflammatory proteins were shared between males and females. However, it is important to highlight some restrictions, such as 3 genomic regions, as well as 2 in the females and the males respectively, where the genetic variants exhibited sex differential effects. Various molecules contribute to the structure and function of an organism, in this case, human beings. Omics refer to the surfaces under the object of study, be it the genome, the transcriptome, or the proteome. The integration of multiple omics layers to understand the biology or the function of an organism is termed as integrative omics. To date, and over the past 2 decades, many disease SNP-associated loci have been identified. To identify the biology underlying these disease-associated loci, expression QTLs have been widely used, yet there is missing link between genes and diseases. In Chapter 6, we show the complementary role of protein QTLs as well, as we observe limited overlap of candida-induced protein QTLs as well as single-cell expression levels. It was also found that the regulation of proteins were found to be cell-type specific. For example, a SNP associated with MMP1 also showed association with other genes, specifically in monocyte, compared to cells like B cells and dendritic cells. Osteoarthritis is a form of degenerative joint disease which occurs when the protective tissue cartilage begins to wear off. People have the tendency to inherit osteoarthritis. Subsequently, a number of OA-associated SNPs have been identified through GWAS. To understand the biology underlying this disease-associated loci, cytokine induced from LPS and basic calcium phosphate, which is kind of crystals mostly found in OA patients, were performed. we found that most of the cytokine QTLs overlap with the OA-associated variants, as indicated in the vertical blue bars. Through the integration of the genome, the proteome, and the transcriptome, we showed that glycerate on chromosome 19 was a driver gene for osteoarthritis. Inflammatory is a common trend in most diseases. In chapter 8 and 9, we profile the all-link inflammatory proteins in sepsis patients as well as dengue patients. In the sepsis patients that developed acute kidney and those who did not, comparative analysis identified 9 proteins that were upregulated in the acute kidney patient. Similarly, among the dengue patients with various phases, we identified a number of upregulated and downregulated proteins. Among the pathways that were significantly enriched for these proteins was the activation of the endothelium, which was shared between both diseases. In short, this study shows that human population genetic analysis requires diversity and ethnicity. Multi-omics approaches also have the capacity to identify disease driver genes, and population-based cohort also can provide insight into diseases. Having presented this summary of my dissertation, I return the floor to the rector.
You have presented a nicely illustrated and understandable summary of your thesis. Thank you for this. And I now would like to introduce the experts who you see are sitting in front of you. First of all, on my left, the 3 members of the supervision committee. First of all, Professor Joosten, he's the promoter and professor of mechanisms of inflammatory diseases at our Radboud UMC. And next to him is sitting Professor Netea, also promoter, professor of experimental internal medicine, also at our university. And finally, Dr. Kumar, co-promoter, and he's assistant professor at the Department of Internal Medicine at our university. These 3 colleagues have supervised our PhD candidate over the last years and have frequently discussed his scientific work with him. Therefore, they will refrain from asking questions today. But that's different to the manuscript committee. The manuscript committee consists of Professor Gielissen, Professor Thierry Serloff, and Dr. Heutgen. And they have intensely studied the PhD thesis and evaluated and given their comments. And they will certainly ask questions today. I would like to give the floor now first to the chair of the manuscript committee, which is Professor Gielissen, and he's professor of genome bioinformatics at our university.
Thank you very much. Before I start with the questions, let me first congratulate you on this thesis that I have indeed studied in great detail. Unfortunately, your thesis is very lengthy. It took me a considerable amount of time, but I must say I've enjoyed reading all of the chapters, and in particular I appreciate your point of studying African populations, which I think from a genetics perspective alone already are much more interesting perhaps than European populations and are severely understudied in our discipline. So congratulations on that. I think this is really a fascinating work that you've done. Let me move on to some of the questions that I had. Firstly, I'd like to discuss with you chapters 3 and 4. And from your chapter 3, I'm actually not so much, I wasn't initially interested in the BCG, I was just interested in the analysis that you did on the control cohort itself, where you, I think, identified 12 significant, genome-wide significant SNPs in your cohort. So maybe my first question, because I'm not somebody who's into association analysis, is that I was a little bit surprised by the high number of findings, given the size of the cohort. I'm typically used to GWAS studies about human length, which require 200 or 300,000 individuals to study. So can you comment a little bit on that, why you think you have so many findings?
Dear opponent, I appreciate your congratulatory messages. And yeah, indeed, in chapter 3, cytokine QTL mapping were performed in the BCG as well as the control group, that is the infants that were not given the BCG vaccine at birth. And indeed, you are right, we identify 13 genome-wide significant SNPs in 12 independent loci. This was also a bit surprising because before this analysis, studies have been conducted in the European population, which is the 500 FG. And then looking at the numbers between these 2 cohorts and the fact that many of these significant cytokine QTLs were observed in the control group, we are also a bit surprised. But what we hypothesize from this finding is that these are just infants who were just born like 4 weeks, that were when the samples were taken. So we assume that, for example, environmental factors, except the internal environmental factors, but the external environmental factors that can also interact with these genetic effects were minimized in children compared to adults, so which could be the reason why we find like an appreciable number of significant cytokine QTL despite the limited number of samples, I think below 200.
Mm-hmm. All right, that could be a very good reason indeed. And then if you compare this to your next chapter, your chapter 4, you have a slightly larger cohort there, and you identify only one significant hit in your controls. So do you think the reason for that is again that these were infants in the West African cohort?
Yeah, I think it could be possible that, as I explained earlier on, these are adult populations, so we could assume that some environmental factors can neutralize the genetic effect, leading to the identification of only one significant loci. But aside that, what I also think is that the African population, as you initially said, is very diverse, and it is possible that the genetic effect that is identified from Guinea-Bissau, which is a West African country, compared to Tanzania, which is East African country, could be different.
Okay, and that would also probably then explain why there's no overlap in the findings that you have.
Yes.
So then, what do you think should we do with these kind of studies? Should we, go through the entire continent of Africa and then, make small subpopulations out of everybody and test like that? Or do you think, because in chapter 4 you also combine Europeans and Africans together and do an association analysis, should we go for just one really big cohort with all of the African continent in there preferably?
I think if it is feasible, I would have wished that this study should be conducted in each of the West African countries, because even physically we see that people from East Africa, people from West Africa, phenotypically we look a bit different. So if it is feasible, as I said, I would be happy for this genetic analysis to be conducted in each of the African populations, because some analyses have shown that there is even diversity within the African population compared to the European population. So people think that the European population are more or less homogeneous compared to the African population. So if, for example, the European population, if you conduct some extensive, as you said, big data or cohort, if you conduct one in the Netherlands or let's say people from Belgium, I actually think it should be sufficient to extrapolate with the other European population, but I think that cannot be said for an African continent.
Okay, and what do you think are we going to find then? Are we, going to find all kinds of different SNPs in the different populations with no overlap, also in completely different pathways, different genes, or is there some convergence going to happen? And how does that compare to European populations then?
Yeah, I think there will definitely be some convergence between the different African ethnicities. I think the fact that there is diversity in the African population doesn't overrule the fact that certain genetic variants can be shared across the populations. But this analysis have not been done, so it will be difficult to conclude that we are going to find like substantial overlap between like East Africa and West Africa. Like the chapter 3 you make reference to, we were dealing with adults against infants. So the overlap will not tell us much about ancestry effect, because we cannot disentangle the age influences on the two comparisons. So our inability to find overlap is not conclusive.
Okay, and now suppose we're going to do this, then what would you recommend? How many individuals do we need from every population if we were able to even define population in a good way?
I think here, for instance, what I think, at least this studies has given us some insight into what we should expect in the African population. So if you ask how many samples, I would say the bigger the cohort, the better. But then we can make inferences from the effect sizes that we identified in these cytokine QTLs, because this is one of the assumptions we need when we want to calculate like statistical power. We should know the effect size of the traits, then we can make an assumption as to the number of samples that we will need for QTL mapping or GWAS analysis.
Okay, do we still have time?
Yeah, for a short question.
Well, you gave a nice introduction to my follow-up question. I was just wondering about these effect sizes, because nowhere in your chapters do you actually, list any effect sizes. Did you look at effect sizes? Can you say something about the variability that you're actually explaining with your findings in these cohorts? Because that will tell you how many you will need, right?
Yeah, that is very true. I think it also comes down with the limitation of these two cohorts. That is why we couldn't compare the effect sizes that much. Because personally, I wasn't comfortable comparing adults and infants, so that's why we didn't go deeper into the effect sizes, because I think the effect sizes, for example, are likely to be higher in, let's say, infant than adult. So even if you compare adult and children, you are likely to come out to some results that could likely be false positive and doesn't represent the true biological effect of these SNPs in the two populations. So if we had a cohort that is comparable between both, comparing the p-values and the effect sizes would be much better to represent the biology underlying these SNPs.
All right. Thank you very much. I give the word back to the rector.
Thank you, Professor Gielissen. Next member of the manuscript committee is Professor Tsiri Serlov. She is professor of hepatic inflammation and metabolic health at the Maastricht University. So welcome here at the Radboud University, and we appreciate that you traveled all the way from Maastricht to Nijmegen. The word is yours.
Thank you. So, dear candidate, I would like to start by congratulating you for your beautiful thesis book, which I also read with lots of interest. I think your work definitely contributes a lot to our understanding regarding the immune component the genetic component on our immunity in both health and disease, and therefore open the door towards future target interventions. I particularly also like the social impact of your book, especially by investigating it in diverse population. I would like to start asking you questions from chapter 5, where you investigated the sex-biased genetics. And I was wondering, because the differences that you observed were not that big. You also said that this is in line also with literature. I was wondering whether you think that this is really the total effect that we see, or is it that if you will take another approach, for example, let's say you will use some kind of trigger to the immune system, you will find bigger differences, and whether this is indeed consistent in different populations.
Yeah, dear opponent, thank you for the congratulatory message. Yeah, indeed, in chapter 5, sex effect on inflammatory proteins were conducted. And as you rightly said, in chapter 5, for instance, the number of SNPs that were influencing the inflammatory proteins between the males and the females were not that substantial. For me, it was very surprising as well, because I think males and females are different in so many ways. And for that matter, I was expecting to find like a distinct genetic variance influencing the inflammatory proteins in the males and the females, but it didn't happen like that. And we have not done this analysis in African population, so it is impossible to also determine whether we're going to find the same effect in other populations or not. So that requires further investigation. And I think the question also relating to, for example, if we have to look at this in different omics layers, I think there's also literature evidence that were performed on transcriptome analysis. So as you said, we discussed that it's in line with literature. So aside proteomics, it has also been shown in transcriptomic analysis. So that kind of gives us a clue that this is likely to be so, or the genetic effects that influence sexes are different irrespective of the omics layer that is being considered.
Okay. But do you think, for example, if you will trigger the immune system somehow, you will find bigger effects there?
I think, yeah, so this was, for example, performed in steady state conditions without any stimulations. So, and if you look at, for example, the immune responses prior to stimulation and after stimulation, mostly we see upregulation of, for example, cytokines and those kind of things. So if there is this upregulation and more variability upon stimulation, then I could expect that the genetic effect will also be different if it is conducted. So, but in chapter 5, this analysis was conducted mainly in steady-state conditions and not in stimulated conditions.
And I was also wondering, why did you exclude the sex chromosome?
Yeah, this was I think one of the issues that a reviewer asked. And I wake up this morning and then this paper was also accepted. So that was the only paper in the thesis that was under review. So it was accepted this morning. Yeah, the exclusion of the X chromosome is, I think it's a complicated issue from biology as well as the technology that is available at the moment, because the sex chromosome compared to the autosomes has some unique features that require deeper understanding before one can go ahead to perform QTL mapping in sex chromosomes. So it is no surprise that it is lacking in terms of the depth of analysis compared to the autosome analysis. And here it wasn't included, but this analysis has been performed. And at a point you have to finish your PhD. So we discussed, and then my supervisor also agreed that we write a separate manuscript for it. Yeah, but I think the reason why it also delayed and it wasn't able to incorporate into this analysis, for example, the imputation approach is very complex compared to the autosomes. For example, the same X chromosome can have like 3 regions. Like the pseudoautosomal region has 2 regions themselves. In that region, the males and females behave the same, and then it's easy to impute that. But if you go to the non-pseudoautosomal regions, a lot need to be done in terms of the quality control processes and stuff.
So I'm happy to hear this is just technical, actually, and you agree with me that probably the impact is bigger than. And if you needed to, because you said yourself that you also think that there is a big impact, definitely in other population maybe, what would be the steps that you will take what will be the implications of such impacts, assuming that you will discover all these differences in health and in disease, for example, in vaccinations or in susceptibility to diseases? How should we take this?
Yeah, I think indeed, if you look at the X chromosome, for instance, the number of SNPs in this region is a bit less compared to the other chromosomes, but they are also important genes in this region. And the lack of analysis in this S chromosome, the clinical implication is that it will be very difficult for us to understand diseases such as genetic disorders, because these are mostly manifested in the S chromosomes. So indeed, I think it is something we need to do in depth and also in different populations, because as you said, if you have to target it in vaccination or therapeutic target and stuff, doing it in one population is not sufficient. So I also agree that in future, if it is possible, analysis on the S chromosomes on different populations is important, because scientific research is leading towards precision medicine, and that requires that each individual or each ancestry is incorporated into genomic research.
So we still have a lot of work to do there. And then, do I have time for additional
Very
question?
short question.
Very short. So in chapter 7, I was wondering, why did you actually use healthy individuals and did not compare patients with osteoarthritis compared to healthy individuals?
Okay. Yeah, indeed. I think in chapter 7, we were trying to understand osteoarthritis loci, the underlying biology. And the question as to why we use it, I think we don't have data on this patient ourselves in order to profile like the cytokines that we use. But we made use of this stimulation effect, and we assume that this can reflect, for example, the disease condition, because as I indicated, one of the stimulations that we used was BCP, and this is a crystal that is found in the osteoarthritis patients. And so it is possible that it can mimic the disease itself. But if you had an osteoarthritis patient, I think the ideal situation is to profile the cytokines from them. That notwithstanding, it is also possible for healthy individuals like steady state conditions, for some of these SNPs to be shared between the disease cohort as well as the healthy cohort.
Thank you very much. I'm satisfied with your answer, and I give the board back to the corrector.
Thank you. Thank you for your participation. Now the word is to the last member of the manuscript committee, Dr. Joosten. He's associate professor at our university in both the Department of Human Genetics and the Department of Internal Medicine.
Thank you, Rector. Dear candidate, also I would like to start with congratulating you on this massive piece and the wonderful work you've done. I thoroughly enjoyed reading it. I learned a lot from it, and I also particularly like the illustrations that you provide. And with that, I also want to congratulate your stellar team your stellar promotion team. So congratulations on that, and thanks for not only bringing GWAS power to Nijmegen, but also some Ghana power to Nijmegen. So really admirable how much extra you add in terms of diversity, and especially we as geneticists should embrace that. So well done. Besides those compliments, I of course still have a few questions. And first of all, I'd like to discuss what you mentioned yourself in your introduction on page 15 and 16, that how to follow up on an association signal. And you have that throughout your thesis, you have that a lot. And I think in the last paragraph of page 16, you write yourself that after finding an association signal, that requires follow-up, and that can be experimental, statistical, or bioinformatics. Just as an open question to start, if you now work on in your postdoc and you would like to follow up on some of your more significant findings here, how would you approach that?
Dear opponent, I appreciate your compliments. Indeed, following up on identified cytokine QTLs is very, very important, because one of the limitations of our study is that it was always difficult to get a substantial number of replication cohorts to validate the finding that we found. So in future, if it is feasible, I think it is good to get another sample of cohort that can be used to, first of all, replicate these findings. After these findings have been replicated and we know that they are robust signals and they are associated with the trait, following up with them is important in so many ways. When I talk about experimental, statistical, and bioinformatics, sometimes it is difficult to perform experiment on some of these identified cytokine QTLs, especially if it falls in the non-coding part of the genome. But if it is in the coding part of the genome, then it is possible to experimentally look at, for example, the nearest gene or something, whether you can perform some knockdown experiment or something to see the influence of the gene on the phenotype that you tested. But if this experiment is also difficult, luckily genomics is advancing, and these days there are many bioinformatics tools online that you can, for example, overlap your cytokine QTLs to see which region of the genome they fall and which category they fall. So you know if they overlap with some transcription factors or some enhancers, then that can give you like a potential function of the identified variants.
Yeah, wonderful. Maybe we do one step back and say, okay, first we have an association with a SNP. Usually that is considered a proxy for a signal.
Okay.
So what does that mean? It basically tells you that the allele itself, the haplotype that you found, may be associated with a certain trait or disease. So what could you do to better understand what on that haplotype is potentially the causative factor or variant? Do you have ideas on that?
Yeah, I think if you find like a SNP, for example, in a certain genomic region, and you want to, for example, come out with the causative SNP itself, I think that is one of the challenges in GWAS, because sometimes you find these genetic variants and it is in high LD with some of the neighboring SNPs and those kind of things. So if, for example, this is highly powered, the data is highly powered, it's been validated, we can perform like a fan mapping to pinpoint the causal SNP itself. And once that is established, I always like the genotypes variation among the individuals. So it is possible if you make a box plot stratified by the SNP genotypes, you are able to know which individuals carries the effect allele or not. So follow-up, you can look at these individuals, if it is possible in longitudinal manner, and see whether they are the ones that end up developing certain diseases or not.
And if I may be a bit provocative, what if the causative SNP is not a SNP?
Yeah,
What could it then be?
I think if it is not that SNP, then likely it's likely to be false association. That can be one. And if it is not the causative SNP, if I understand you correctly, maybe we are talking about the top SNP that were followed up. There are instances where the top SNP itself is not the causal SNP itself. So if it is not, then maybe it is better to investigate other neighboring SNPs, and probably those who end up to be the causative SNP and something like that. But for example,
I was pointing more towards another variant type. You write that also in your introduction. So of course we have more than just base substitutions in our genome.
yes.
For example, structural variants may have a much bigger impact on almost any locus in the genome. So what if the proxy SNP that you find and replicate, as you clearly demonstrate or explain, what if the actual causal variant is actually a structural variant?
Okay, I think if it's a structural variant, I don't think we studied other kind of genetic variations in this disease, with the exception of single nucleotide polymorphisms. But it is also possible for structural variants to have impact on these traits, cytokines or inflammatory proteins that we profiled. And to look at the effect, I think first of all, it is important to study the nature of which structural variant it is, whether there was duplication or there is substitution or deletion of the nucleotide base pairs. Once that is established, then I think that can give you a clue about some follow-up analysis that can be performed.
Great. Okay, then maybe we switch gears a little bit, although it still touches on that. In chapter 6, you describe some more cell type specific effects, and you're right. I think a lot of the immune traits and potentially also immune diseases may actually be cell type specific. And for a genetic variant to convey a cell type specific effect, there's only a few possibilities how that can happen. So it could be the gene itself that is expressed only in that one specific cell type. But how could you think of a regulatory effect that may be cell type specific? Which genetic element needs to be hit for that?
So if, for example, as you said, if you are looking at the regulatory regions, for instance, and for example, which cell types are likely to be involved, I think we first need to look at which part of the genome does the SNP overlaps with the regulatory elements. For example, if it is in the promoter region or not, or the enhancer region and stuff. And that can give you a clue about which cell type to look at. But I think looking at the cell type effect in that region of the genome can be a bit challenging.
All right.
Compared to, for example, if it falls in the coding part of the genome.
All right. I'm very happy with this discussion, so I'll give back the floor to the rector. Thank you very much.
Thank you so much. At this point in time, I would like to thank all the members of the Manuscript Committee for the hard work. Your contribution is greatly appreciated. We move on via the online participant, which is Dr. Jonkers. He is Associate Professor of Immunogenetics at the UMC Groningen. The word is yours.
Thank you, Rector Magnificus. I too would really like to congratulate you with this incredible piece of work. As an immunogeneticist, I'm really intrigued with the variable responses of cytokines to all sorts of stimuli and how big the role of genetics is in this. And I would like to congratulate you with this impressive piece of work, but also your supervisory committee with this achievement. So as an immunogeneticist, I'm actually more interested in autoimmunity versus infectious disease. But I think that the responses that are associated with infections definitely also have an effect, of course, on autoimmunity. So in your introduction, for instance, you mentioned that there's 2 examples of genetic variants that are selected for as a consequence of infection. And these are specifically ARAB2 and TIC2. I would like to ask you, can you reflect a little bit on the interchange between genetic selection to infections and autoimmune risk?
Dear opponent, I appreciate your compliments. Indeed, genetic effect, or for example, let's say mutations in these genetic variants can lead to autoimmunity, as well as can also influence infectious diseases. But in terms of the risk, I think, for example, the effects or the magnitude of infections can be manifested, first of all, if the immune system is kind of diminished in some way. So if, for example, there is excessive inflammation, or there is excessive, for example, inflammation, so that can really cause, for example, the immune system to diminish. And that will pave the way for infections to also develop into the patient or the individual that is having like a weakened immunity.
But in autoimmunity, what you often see is that there is an increased immune response towards the self-antigens. So how is the interaction there, do you think?
I think in terms of the interaction in autoimmunity and, for example, the interaction that you talk about, I think it is a complex or dynamic interaction between the host itself or the host genetics, as well as the infectious pressure that the host is being exposed to. So if, for example, the host is being exposed to various infectious pressures, then we expect the interaction with host factors to be more advanced compared to if, let's say, the individual is exposed to lesser or, for example, no infectious pressures. So to answer your question, I think is the interplay between the host as well as the host factors like genetics, or it could be the age or the sex, as well as the kind of infections that the patient is being exposed to. So if, for example, in studying like infections, we also try to use like various stimulations, as you alluded to. And I think these kind of simulations already have a different magnitude on the impact, for example, of cytokines that we measured. And the fact that these kind of simulations have different impact on the cytokine responses, then I could speculate that, for example, that will happen the same way depending on the kind of infections that, be it viral, bacteria, or whatever that the individual is being exposed to.
Okay, thank you. So to follow up a little bit, so there has been an infection and an increase in autoimmunity in the last 50 to 100 years. So incidence of autoimmune diseases seems to be going up quite significantly. So if there really is a link between infection and the risk of autoimmunity, then arguably the selective pressure of infections could also increase the autoimmune risk. So what do you think about the healthcare and how treatment options with regards to vaccines, better hygiene, and antibiotics has as a consequence on the selective pressure of infectious diseases on the genetics?
Okay, I think if you look at selective pressure, for example, I think it's one area that has not been extensively studied, because, for example, there are regions that have regions of the genome that has undergone like natural selection already, and it was assumed that this is basically in Europeans. But these days, I think in the discussion or somewhere, there were studies also from people from Ethiopia, that we found like new genomic regions that have undergone like selection already. So first of all, I think that aspect need to be properly investigated to be sure, because, for example, toll-like receptor that we investigated in chapter 4, it were thought to be, okay, this is European, and likely we didn't find it in the African population. But I think there are regions that can undergo natural selection in both populations. So if you're able to understand this, that can influence decision of the vaccines. Then we are able to target specific regions that we are sure that the natural selection has been occurred in diverse populations, and they will be more representative to target for vaccines, probably compared to regions that have undergone like natural selection in specific populations.
Okay, thank you. I'd like to ask you something also about page 116, chapter 4, Q. So one of the things that I was really struck by is that the interferon gamma response as a consequence of stimulation with different bacteria seems to be much more variable compared to the other cytokine responses. And I was wondering, could you elaborate a bit on why you think that this is the case?
I think the biology underlying these various cytokines, interferon, TNF alpha, and the rest, I'm not privy to the details biology underlying all these cytokines. But then if you see, for example, interferon is more pronounced compared to the other cytokines, it is possible that that is the biological nature of interferons. Also, it could be that the kind of stimulus that we selected, they are able to induce more interferon compared to the other populations. But that being said, I think interferon has been associated with a lot of viral infections. And for example, the population that we targeted in chapter 4, it is obvious that they have been exposed to various infectious pressures. And for that matter, it is possible that due to that, the interferon is also more pronounced in the population itself. So I think it could be due to the biology underlying all these cytokines, and probably the stimulation that we use are able to induce more interferon, or it could be the selective pressures that the people from Tanzania have been exposed to that leads to the abundance of interferon. As I said, it's been associated more viral infections, and I think it's one of the continents that is noted for some of the viral infections like HIV and stuff.
Dr. Jonkers, I hope you're pleased with the answers.
Yes, I am. Thank you very much. I give them.
Okay, thank you for your contribution. We move on to Dr. Vermeulen. She is Associate Professor in Genetic Epidemiology at our university.
Dear candidates, I would also like to start with congratulating you and your supervisors on finalizing all the work that is described in your thesis. It's a lot of work. It's a massive book. And like I said earlier, I also applaud your efforts in creating more insight into the overlap and differences between a Western European and African population, and also in differences with regard to sexes in the underlying genetics. And my first question is related to that. And I want to discuss the results that you've described in chapters 3 to 5, in which we evaluate these ancestry and sex differences. And you show that there is actually minimal overlap between African and European cytokine QTLs. It's actually really low. It's less than 2%. But the overlap of sharing between males and females is actually quite high. And I was wondering, of course, the results could be due to actual underlying biology, but it could also be due, the very low overlap in chapters 3 and 4, to other issues or alternative explanations. And I was wondering whether you could please name a number of aspects related to study design and analyses that may have potentially led to an underestimation of the shared CQTLs for the African and European population.
Dear opponent, thank you for the congratulatory messages. Indeed, in chapter 4, prior to that, I think in chapter 3, we also tried to compare the cytokine QTLs between different ancestries. But the analysis were more compelling in chapter 4 because the two populations were somehow comparable. Both were all adults. And indeed, we found very limited overlap between the cytokines that we considered between both groups. I must admit that some of the cytokines were not able to be investigated because we didn't have all the cytokines that were profiled in the European cohort and the African cohort being common. So only 12, for example, cytokine stimulation pairs were interrogated, and we found a restricted overlap between the Africans and the Europeans. So for the other cytokines, it is not clear how the effect will be manifested. But to me, it is not surprising that we found a limited overlap between the Africans and the Europeans, because clearly the genetic architecture between these two populations are different. And we showed that, for example, different haplotypes, I think in one of the figures, we found like the haplotypes in the European populations were higher. Haplotype length and the number of haplotypes were higher in the Europeans compared to the Africans. So that might be the reason why we find the limited overlap between the Europeans and the African populations. And many factors could also explain that. And I can also think about like a founder effect. So founder effect, what I mean, for example, it is obvious that contemporary human beings originated from Africa. And for that matter, people who move to different countries, whatever, can form like smaller communities. Mm-hmm. And for that matter, the genetic effect will be a bit different compared to the founder itself. Mm-hmm. So that could be one. Yeah. And coming to chapter 5, where...
Yeah, or maybe go back to the design of your studies, if there's something in there that could have explained the limited overlap. Could you reflect on that? You mentioned differences in the study population with regard to age. You already mentioned that, but is there something different or something that you could have done better, or you think that may have affected your results with regard to study design?
I think you talk about chapter 5 as well as chapter 4. Mm-hmm. So I will look at the study design from chapter 4 first. For me, chapter 4, what I think was, for example, I've already explained that in terms of the study design, the number of cytokine stimulation pairs were not comparable. That is one. And then second, if you look at the study design, I will see, for example, the limitation of the absence of some of these stimuli there. And second, if you look at the genetic data itself, different SNP arrays were used to profile the genetic variants in the Tanzanian population as well as in the Dutch individuals. So from that angle itself, we are already losing some markers, because if you overlap, as I remember, like after imputation, there were 5 million SNPs in the Tanzanian population and 4 million in the adult population. But if we had to use like a common array to genotype these two populations, it is possible that we might have a higher genetic variant that are shared between both populations before it is being tested to check whether the effect will be manifested or not.
Yeah, I guess that's a differential misclassification error that you make between the populations. Yeah. But even more simply, if you look at chapter 5 versus 4, there's something different there. When the timing of meta-analysis is different, right? So one of the things I think is, you mentioned replication as one of the important things that you would like to do in future research. So if you think about chapters 3 and 4, have you done replication prior to comparison of the findings?
Yeah, chapter 3 and 4 compared to chapter 5, I think the study design, for example, in chapter 5, we made use of inflammatory proteins, and those were steady state conditions compared to chapter 3 and 4, where induced cytokine QTLs were profiled. So the study design is a bit different, because I would expect that stimulated conditions versus unstimulated conditions, given that we see pronounced effect upon stimulation, the genetic effect will also be different. Yeah. So if you have to compare chapter 3 and 4 against chapter 5, that is one of the differences I could say. One is stimulated and one is not. Yeah. And indeed, chapter 4, one of the problems is that is why meta-analysis were performed by combining the two cohorts, because the Olink data were profiled at different time points. So it's likely to be, there's likely to be batches or false association based on the timing of the profiling of the Olink inflammatory proteins. Yeah. So if they were profiled at the same time, as you said, in terms of cohort size, then probably we could integrate the two, because they were both from the same population, like 300 FG.
And what I liked about chapter 5 is that you first...
I hope you're convinced by his answers.
Almost. Okay, yes. Thank you. Thank you for your answers. And
thank you so much for your contribution. Yes. We move on to Dr. Matsaraki. She is postdoctoral researcher in biostatistics at the Department of Internal Medicine. The word is yours.
Thank you. Dear candidate, first of all, I would like to give my compliments to you, your promoters, and co-promoters. I really enjoy reading this book. It's a very nice work with interesting findings and new insights. Congratulations. So today, last but not least, I would like to take the opportunity to ask you a few questions for my understanding. I would like to start with chapter 3 with some specific questions. So on figure 5A on page 66, you observe a significant enrichment of cytokine QTLs in the BCG vaccinated group at several complement genes. This is a very interesting finding. You tested different windows between 50 and 250 kb, and a striking significant enrichment in complement genes was found when using a window of 150 kb. This significant enrichment is lost when using a slightly bigger window of 200 kb. So can you help me understand this analysis, and how do you see a significant enrichment at a window of 150 and not a little bit slightly bigger window of 200 kb?
Dear opponent, I appreciate your congratulatory message. Indeed, in chapter 3, we perform the analysis leading to that figure is a competitive gene set enrichment analysis, and that was triggered based on the identification of one of the significant cytokine QTLs falling within the complement genes, as shown in one of the regional association plots. So the idea was, if one of the genetic variants falls within the complement gene, then it is possible that other genetic variants can equally do so. So how the analysis was done was first to map the genetic variants, in this case the cytokine QTLs, or the significant ones, to various complement genes. So this was like overrepresentation analysis. So what is the proportion of the significant cytokine QTL that falls within the complement genes compared to the number of cytokine QTLs outside complement genes? And indeed, various window sizes were tested. And if you look at the result, you are right. It seems that the analysis is really sensitive to window sizes, because at window size 150, we see significant enrichment, and also with 0. These were 0 kb. But I think, even though it looks like the result is sensitive to window sizes, I was happy to see that at window size 0 kb, there is also significant enrichment as well. In that case, what it means that the top SNP that we identified, if we are considering like 0 kb, the SNP is falling in the complement gene of our interest. And these results are based on the SNP p-values. So if you don't find, for example, this significant enrichment with other threshold, then what it means is that, let's say some of the p-values, if you increase the window sizes, are very smaller or weak compared to the one close to the top SNP itself, because that is where we identify the significant cytokine QTL. So the effect for that one is likely to be higher compared to the other SNP in the neighboring region of the complement gene. So why we see the different effect, I think the result seems to be sensitive to window sizes, but since the analysis is based on SNP p-values, if you do like the combination of weaker and higher p-values, then it's possible that regions where it balances will not be significant at all.
Right. Thank you for the explanation. I will also be happy with these results. Right. So I will follow up with another question in the same chapter. So in addition to the enrichment analysis, you also made use of publicly available ChIP-seq data from CD14 plus monocytes, and revealed that the cytokine QTL identifying the complement locus is within an enhancer region that interacts with neighboring genes. So you looked at the epigenetic regulation in CD14 plus monocytes, and therefore you next chose to do knockdown experiments in monocytes, as seen in figure 5D on page 67. So these experiments showed that the depletion of complement genes results in a significant reduction in interleukin-1 beta transcription. And we know that trained immunity, by definition, is an enhanced response upon a non-specific stimulus of the innate immune responses. So my question is, what would be your conclusion based on these experiments? Do you intend to conclude that the complement genes are detrimental for trained immunity?
I think I will answer from where you ended. To say that it is detrimental for trained immunity will be a bit premature, because this analysis, we didn't look at trained immunity itself. We just look at the stimulation experiment and stuff. And indeed, if you look at the figure that you referred to, you could see that if you, for example, knock down the complement genes, the cytokine, in that case, IL-1 beta expression were also reduced. So this kind of tells us there's some kind of relationship between the complement genes as well as the cytokine, in this case, IL-1 beta. That is why I'm not entirely sure that immunity will be detrimental, because I think if it is depleted, maybe the effect is downregulated. And if it is downregulated, it can also have some biology compared to trained immunity, where you are expecting like enhanced responses following like subsequent stimulations and stuff.
Right. Thank you for your answer. But this cytokine Q12 was mapped after BCG vaccination, right?
Yes.
It's in the context of trained immunity. All right. I would like to move to chapter 5, where you looked into the sex-specific regulation of inflammation. In this chapter, it is acknowledged that the chromosome X is missing from all the analysis, and it is stated that chromosome X accommodates the largest number of genes related to the immune system. Would you expect an enrichment of PQTLs in the chromosome X compared to autosomal regions? What are your thoughts? Oh.
I think
The defense is hereby concluded. I give the floor once again to the candidate.
having defended my dissertation to the best of my knowledge, I would like to thank my director, my supervisors, as well as my co-supervisor, as well as all those who have honored this ceremony with their presence.
The Doctoral Examination Board retreats for its deliberations. On behalf of the Doctorate Board of Radboud University Nijmegen, we have decided to award you the degree of Doctor. I hereby invite Professor Joosten to discharge the task assigned to him.
With the power entrusted by law to the Doctoral Board, I hereby confirm upon you, K Boahen, born in Quintabo, the title of Doctor from Radboud University Nijmegen, to which attach all associated legal and customary rights and duties with respect to academia and society. As proof thereof, I presented you with the doctoral diploma signed by the rector and the doctoral thesis, supervisors and co-supervisor.
Please be seated. The laudatio will be spoken by the co-supervisor, Dr. Kumar.
Dear Collins, it's a great honor and pleasure for me to stand here in the capacity of a co-promoter to deliver the laudatio. Before I explain to the audience who you are and how you got this far, you will be Dr. Boahen soon. I would like to congratulate you and your family, with your forthcoming doctorate upon successfully defending your thesis. According to your CV, you were born in Kintampo, Ghana. You obtained your BSc degree in biomedical engineering at the University of Ghana. After that, you decided to move to South Korea to pursue your master's in biomedical engineering at Hanyang University, South Korea. Here, you embarked on an interesting field of imaging genetics to quantify the heritability of human brain network, which also provided you with the first opportunity to make yourself familiar with some of the important genetic concepts that you later applied in your PhD research. It will be surprising for many to hear that your next choice after your master's was New Zealand, where you wanted to pursue a PhD. You actually had gone through all the necessary procedures required for PhD admission there, but somehow in the last minute there were some limitations on funding possibilities. Nevertheless, Professor Tony Merriman at the University of Otago was so impressed by your expertise and passion that he immediately put you in contact with Leo to explore PhD opportunities in our department. Now that's how I got your CV and came to know about your interest and passion to understand the impact of genetic variation in different human phenotypes. I remember still your interview with four of us where you showed a lot of maturity and made a very impressive presentation on your master's work. Motivated by these initial interactions, we were extremely happy to offer you the PhD position in the group to explore the genetic regulators of immunity. When you joined the department in 2019, you were not only familiar with genetic concepts, but also motivated to apply statistical knowledge that you had gained from your bachelor's and master's studies. You were not formally trained for immunogenetic analysis, yet you were very much willing to learn immunological concepts, although at times it was difficult and complex for you. I still remember all the lengthy discussions we used to have on these topics, especially trained immunity, in the beginning of your PhD. The content of your thesis clearly shows that you were extremely motivated to take up the new challenges during the entire PhD career. We are very impressed how you were eager to go out of your comfort zone to tackle these diverse projects. These topics ranged from traditional genome-wide association studies to quantitative trait loci mapping, X chromosome imputation, although it couldn't include that in the thesis, but you explored it in detail, to population genetic studies, single cell expression analysis, to multi-omic integrative studies. Now, handling such diverse topics also demanded you to collaborate with multiple researchers in the field. For example, you also visited Professor Franke's lab at UMC Groningen for a couple of months to learn more about single cell RNA sequencing, EQTL mapping, and so on. Collins, you're one of the most ambitious and hardworking people I have ever met. I'm sure many in the group agree with me when I say that your passion and dedication to perform impactful research is commendable. You were very independent in planning your analysis, yet extremely collaborative and eager to help colleagues and partners. This was very important for us as we were collaborating with so many groups and so many people from all over the world to deal with different topics and concepts. Although you found some of these collaborations to be problematic at times, I really hope that together we were able to learn a lot from these experiences and that you will benefit from the advice that we gave while you embark on your postdoc career. Now, apart from dealing with diverse projects, you also had to slightly modify your thesis goals during the pandemic time. I know that it was challenging for many and also to you, but you were so determined that you actually sacrificed many hours of your late night, weekends and holidays to really fulfill your goals, which resulted in this beautiful thesis that you successfully defended today. Collins, I should also mention that you showed a tremendous progress in your scientific writing skills in a very short time. This was one of the minor concerns we had when you actually started your PhD. For example, your first paper needed quite a few rounds of revisions and editing. However, you were able to grasp many essential points about drafting a manuscript quickly, and you managed not only write clear manuscripts, but you were also able to generate very strong rebuttal letters, which I appreciated a lot. I'm also impressed with improvement in your presentation skills. I'm sure many in the audience also agree that most of your presentations were very informative and educational. In addition, you received several invitations for oral presentations in international conferences, and recently you were also invited to chair a session on one of the prestigious international immunogenetic conferences. Now, these qualities of you clearly indicated that you are a very positive person and not scared to take up new challenges. However, on a lighter note, the only thing that made you remain skeptical and reserved is Dutch food. Although we all tried our best to convince you to be a bit more open to local cuisine, Dutch sandwich and salad never stood a chance against the tasty Ghanaian food. Now, because of this, in many of our lunch meetings, you ended up buying a bottle of chocolate milk, as lunch, instead of looking at sandwich and different varieties of bread. Collins, your extraordinary talents are not just limited to science. I hear that you are a fantastic cook, a skillful soccer player, and a crazy fan of Chelsea Football Club. Being an illiterate myself on football, I couldn't contribute much to the discussion around that, during our discussions, but I'm sure you found many crazy fans like you in the department to share your joy and frustrations over your favorite club. Collins, many colleagues point out that your perseverance, intelligence, strong personality, and respectful manner stood out from the rest. I couldn't agree more. You were also a very kind and helpful colleague to many in the team. Apart from supervising master's internship projects successfully, your commitment to help and share your knowledge with fellow PhDs, even in the midst of your busy schedule, is greatly appreciated. I'm sure these interactions had a very positive influence on many members in the team. And I'm also sure that your family today, particularly your father, brother, and sister, are very proud of you with what you achieved. Collins, it was a pleasure to be your supervisor and a colleague all these years. But I would also like to emphasize the role of Professor Mihai Netea and Professor Leo Joosten, without whom your supervision would have been incomplete and short-sighted. Collins, you had been an excellent colleague and a fantastic friend to many in the lab, and we are very happy that you will be staying with us for a few more years. Mihai, Leo, and I are extremely proud of you, and you should feel thrilled with what you have accomplished with your thesis and your defense as well. We wish you wholeheartedly all the best for the future. Having said this, I would like to give back the word to your promoter.
Thank you. I too would like to congratulate you very warmly, also on behalf of the doctorate board, for obtaining your PhD today. And of course include in my congratulations your family members, your colleagues and your friends. And I must say a very special congratulations to your partner, your father, your brother and sister. Very special that you all are here today, and it must be a very special day for you as well. You can all relax now. We all witnessed that Dr. Boahen did very well and did get his degree. Dr. Boahen, your research has provided important new insights into immune variability in diverse populations. I wish you every success in your scientific career and all happiness in your personal life. Before we will close this ceremony, I would like to give Dr. Jonkers also the opportunity to say congratulations to you, because she is not able to shake your hands physically. The word is yours.
Dear Dr. Boahen, dear Collins, I'd like to just emphasize how impressed I have been with your, while reading your thesis. The breadth of work that you've presented with the number of stimuli, vaccines, bacteria, and the responses on those in diverse populations from Guinea-Bissau to Tanzania all the way to the Netherlands is truly impressive. And I think that you have shown incredible capabilities by analyzing and working with all these different large, impressive data sets. I wish you all the best in your future. I'd like to also extend my congratulations also to your family and your friends and partners and colleagues, and of course also to your supervisory team. I'm sure that there is a bright future ahead of you. Hearing the comments from Vinod, I think that you will have no problems in finding your way in an academic setting. I hope that you're going to have a beautiful, wonderful, and very festive day. And I say cheers to you, and I hope that you'll have a lovely celebration tonight.
Thank you, Dr. Jonkers. Very nice words. We have now come to the conclusion of this ceremony, and I ask you all to stand. We close with the following words: Gratias tibi agimus, omnipotens Deus, pro omnibus beneficius tuis, qui vivis e regnas per omnia saecula saeculorum.